We'll get started here in 1 second. I'm assuming YouTube's going to start talking again. Oh, man, maybe I worked it out. All right, good morning, everybody. We will get started with this stuff. I hope you've come with some questions that you are hoping to get addressed here. I'm screen sharing the chapter right now, which I'm intending to scroll through and show a couple of different things as we move through here. For those of you at home, I also have opened up the study guide, so I do have that in front of me, but I can't share both. At least I don't think I can share both simultaneously. If that's the case, I got more learning to do up here. Any questions? I'll give you a second at home. Try to be watching this stuff. Yes. Oh, okay. Okay, so the question, this is the second page of the study guide. What is an MIC? Kirby-Bauer disk diffusion and E-test, and then are they qualitative or quantitative? So with that, this goes back to, it would be super awesome if I could just tell you which chapter this came out of, but I can't right off the top of my head. That doesn't matter. So some of the antibiotic susceptibility stuff is what we're thinking about. So oftentimes, I'll give you just a quick spiel real fast. Oftentimes we think about diagnosing an infection, we cultivate, we isolate, we do some biochemical tests to figure out what we're really dealing with. And then the next step would be, what's the drug of choice? How do you know what you're going to treat with? And potentially the concentrations, the dosages that might be effective with that. So an MIC is a minimum inhibitory concentration. That is a dilution of antibody, or not antibody, a dilution of whatever antimicrobial agent. All right, so some sort of antibiotic, not antibody, some dilution scheme is set up. Usually it's a twofold dilution as we mix handfuls of these tubes and dilute out that specific drug. And usually you've got handfuls of different ones you're testing simultaneously. So racks full of tubes that have your bacterium that you're hoping to fight, that you've got a turbid suspension of that you put into each of these tubes, and then a specific amount of antibody. So you're looking at these tubes and you should see basically high concentrations of antibiotic are going to kill off those organisms till you get it diluted out enough to where those tubes look cloudy, they look turbid, so it's not effective. And the tube right before that, where it's still clear, would be your minimum concentration, the lowest amount of that drug of choice that's effective at killing or making it at least look clear, because technically in a circumstance like that, you might plate some of that out and see that with the MIC, there's still possibly something that's viable in there. Clearness in the tube. Typically, we think it killed them all, but you know, deep down, maybe that's not 100%. And then that's where we look at the, you know, the maximum amount of killing with more of that. And we're, you know, it's MIC we typically think about, not the, what was that, MBD. Doesn't matter, all right? So MIC is when you do those dilutions, that's definitely a quantitative test because you can get a specific amount, right? The Kirby-Bauer disc diffusion has a specific amount of antibiotic impregnated into little filter paper discs. Those are placed onto a petri dish that has a lawn of bacterial growth. So bacteria's been spread out all over the plate. The discs are pushed onto the plate and they diffuse out into the medium. As this happens, if that bacterium is sensitive to that antibiotic, it's going to kill it. And so you'll see a zone, but it's a finite amount of antibiotics. So Kirby-Bauer is a qualitative test. It only shows a yes, no, this antibiotic is effective or not. It doesn't give you a dilution or a concentration that might be effective that we might think about, you know, for the patient. The E-test is sort of like a Kirby-Bauer. You've got a strip of antibiotic that has a dilution of drug on that strip. So now it's serving like Kirby-Bauer, where it's a filter paper thing that's placed onto the lawn of growth, but it's also an MIC because you've got the antibiotic diluted in that strip. So the E-test is quantitative as well. MIC and E-test quantitative. Kirby-Bauer is just qualitative, just a yes, no, all right? And it depends on the circumstances. Sometimes Kirby-Bauer is sufficient just to see which drug is ultimately effective. Other times, you really want more details as far as what you might be utilizing. Is that good? Perfect. All right. Next question. Let's see if I've got anything up here. Oh, there's chats. Okay, oh, modes of action of antibiotics. That truly, I want to just flip to the image with this. Mechanisms. So you should be seeing this figure with the bacterium in here, just you know, we talked about this before when we showed what's up with the bacterium. I don't like how it's looking almost as if the chromosome is linear because really they're circular. That doesn't matter. We're not talking about that. So this is what you want to think about. You don't need to know the specifics with the drugs, but antibiotics function by interacting with or inhibiting the synthesis of cell wall material, getting in and basically making it so that the peptidoglycan cross linkages don't form properly is one example with some of that cell wall inhibition. The plasma membrane, there's a handful of drugs that get into this lipid bilayer and cause issues, permeability issues that sets the stage for lysis of the cell. There's other drugs that function with like the protein synthesis, and there's a handful of different ways that they really work, as you see here, targeting different regions on the ribosome, but basically inhibiting the production of proteins, so messing up the translation process. Bacteria synthesize all sorts of stuff. They make a lot of things on their own. Some of these things, like folic acid, are awesome targets for bacteria because we get folic acid from our diet. So taking a drug that inhibits folic acid synthesis does absolutely nothing to us because we're not making it, all right? Metabolic pathways, if you can drive a wedge in there, that might be something that's pretty specific to a type of microbe and not to us. And a lot of times when we think about some of the antibiotics, issues that they may have with us are definitely a consideration. So if you can minimize some of that stuff, that's a benefit. Lastly, are the replication of its chromosomes, so DNA synthesis or RNA synthesis. So before we're ever targeting the ribosomes, we might target mRNA production, and all of those are mechanisms that, you don't need to know all of the specific drugs, just that these are all ways that we may target. And it's not, as far as the test goes, there's not a huge amount of stuff that really focuses on this in the first place. Just something you should recognize. There's multiple ways that we might target organisms. And when it comes to other things like fungi, there's some similarities as far as ways that we may go in and try to target. It's not the same types of drugs for the most part, but there are similar modes of action when we think about that. And we're not, we don't need to get bogged down with that. Okay, let me see if I got another chat up here. Hopefully that answered your question. Anything else from here? I know there's more questions. Okay, let's see, I want to show you, not that this isn't good here. I want to go to chapter 18, I think. There's a couple of things that I felt like were pretty good to kind of just review the littlest bit. This figure, 18.2, shows, and it's kind of arbitrary stuff. We talked about this a little bit. It's concentration antibody on the y-axis, time on the x-axis, but we have no real specifics with this. You've got your initial exposure to some foreign substance. This could be the real deal, a real bad guy, or it could be a vaccination, right? One way or another, all right? So this initial exposure, you see the production of an immune response. There's another figure that shows the immunoglobulins IgG and IgM, and really what's happening with both of those antibodies over the course of time. But this sort of averages the two together and puts it just in one figure, all right? You see an initial response, the primary response, there's the production of those immunoglobulins. It takes typically a couple of weeks before you start seeing this primary response. It starts to decline shortly thereafter. And at this point, we see a secondary exposure. So another encounter with that substance. And that response is much more rapid and considerably more robust. You can see it almost tripling the level that we saw with that primary response. And this is important when we think about truly mounting a viable or a good immune response to fight off these foreign invaders, right? So it could, as we said, initial exposure and secondary exposure to a true pathogen. We can think about, and somewhere in this study guide is sensitizing and shocking dose. That's really thinking more along the lines of allergic reactions, but it fits with this as well, all right? And you can think of that sensitizing dose as this initial exposure. That first time that you've encountered it, you've been sensitized to it, all right? And it's that second exposure that we would call the shocking dose, when you are stimulating these cells to respond, all right, after they've seen it. And it could be similar. We could think about vaccination and your first shot and then the booster shot, that because typically vaccines, usually we think taking multiple shots. that booster shot would be a second exposure, and it would boost up or increase the level of antibody that you've got circulating around to definitely, in those circumstances, enhance your protection. That's the idea with that. And I've got more that I'm going to scroll down to, but I just, as I got on here, I wanted to talk about that. All right, MHC1 and 2, and how do they relate to complement proteins? That's a good question. All right, probably one I asked on the study guide, I bet you. All right, so the MHC1 and MHC2 proteins are a class of proteins that are expressed on our cells, right? MHC1 proteins are expressed on all of our nucleated cells, all right? So basically, all of our body cells, except for the erythrocytes, your red blood cells, they're non-nucleated. They don't have MHC1 proteins on their surface. They have some other stuff, some glycoproteins, some sugar protein combos that allow for the ABO blood grouping system. That's another question, all right? The MHC1s are our own cellular or molecular fingerprints that say these are my body cells, all right? MHC2 proteins are found on some of our white blood cells. Macrophages and dendritic cells are good examples of that with the MHC2s. So they are responsible in the presentation of foreign substances, all right? So a cell recognizes this foreign invader, phagocytizes it, processes and presents those antigens, and it presents them on the MHC2s. So it's a mechanism for these cells to communicate with other cells and basically kind of do like a show and tell situation to say, look at what we found, right? As far as their relation to complement proteins, ultimately what's happening with this, if we see an immune response recognized by those cells on that, those antigens, excuse me, on those cells that have the MHC2s, that can stimulate an antibody response that can activate complement proteins. We can also see other issues with our own cells where the MHC1s are non-functional, if you will, and that can stimulate more of like a cell-mediated cytotoxic type of a response. And I don't think complement really plays a role in that, but that's the idea with that. Questions about that? And there is, I think it's right in this chapter here in 1 second, a little bit of detail about the phagocytic process, a dendritic cell showing that, and I will hit that in 1 second. As I scrolled right to this, I want to just mention this quick anatomy of an antibody or an immunoglobulin. There is a table that talks about some differences with these guys. We'll hit that here in just 1 second, all right? But real quickly, this table, as we look at this, you can see in purple is the heavy chain, all right? In green, the light chain. These chains are linked together by disulfide bonds, so each of these separate chains have some cysteine amino acids. Those cysteines adjacent to one another will form covalent bonds, making a strong structure, all right? Next door to it is a ribbon model showing some of this, and you can see a little of this linkage in between. The heavy chains are also linked together, holding it, again, keeping the structure, if you will, all right? There's a question in here about, the antibody structure. This is what we're looking at right here. So figure 18.5 that details it. Heavy light chains, the FC region is the tail of the immunoglobulin, the end of the Y, all right? This, with some antibodies, is what's going to be poking out after they've linked up to foreign cells. some antibodies, all right? And we will hit that here in just a second, all right? So the FC region is important in stimulating other types of reactions or being recognized by other cells as like a red flag, if you will, and we need to see what's going on with these guys, all right? So the FC region, the FAB region is the antigen binding region, F for fragments. And so this binding region, there is a bit that is constant. So it's all the same. These solid rectangles, these are constant regions where the amino acid sequence is all basically the same, all right, providing this framework. It's the tips of the Y, all right? That's the variable region. These that look kind of irregular, all right? And this is where we see huge variation within this variable region, as the name implies, all right? There's all sorts of different assortments or arrangements of those amino acids that gives a three-dimensional shape to this molecule. And that three-dimensional shape that they confer provides this binding site, this antigen binding site that will recognize a specific epitope, a specific fragment of an antigen will correspond to this region, this variable region, and it will link up. The picture before shows some of this stuff happening right here. So you've got a handful of different antibodies or immunoglobulins for these respective epitopes, all right? Greens, reds, and yellows, all right? And each of these weird shapes on the ends of your Ys would be that variable region, right? Specific for these respective shapes. And they only link up with their like ones, all right? And the idea would be to take these guys out of commission, to cause them to clump up or agglutinate is one potential that they may do, all right, in this reaction. One, all right? So there's your antibody. Here are the different classes. There's a little tidbit about each of them, and then the table. And this, I recommend you look through this a little bit. There are a few details in here that I want to share with you quickly. If I can get it so that it's showing up on the screen here. So the specific immunoglobulins, there's 5 different classes: IgG, IgM, IgA, IgD, and IgE, all right? Little bit about each of these that are important. There's a lot of business on this table, but there are only a few things on here that you really kind of need to focus on, all right? The way they appear is kind of important. You can see there are a handful that are monomers, so just a single Y, all right? IgM is important. It's a pentamer, 5 linked together, all right? And IgA is also a little bit different. It does exist as a monomer, but when we think about when it's actually really doing its job in mucus secretions, it exists as a dimer. So 2 of them linked together, all right? And there's some importance with that. IgD is on the surface of B cells, all right? And that is important for recognition of foreign substances and activation of those, basically stimulating them to convert into plasma cells, all right? So with that, you can see the potential for binding to antigen, and it's all based off of the antigen binding sites up here, all right? Sizes you don't need to worry about, percentages you don't, but you do want to recognize IgG is a dominant player. It's IgG and IgM that we typically think about as the main players in immune response, and IgG dominates, all right? IgE is very low unless we're talking parasitic or allergic reactions, and then you would see the levels of IgE considerably elevated, all right? And that is a red flag for issues, all right? I do see there's a chat here, but just give me 1 second and I will hit you. All right, so these 2, really these next 3 in this table are important, all right? IgG is the only immunoglobulin that is capable of crossing the placenta That's important to provide immunity to that child before it ever hatches out of its mom, all right? That's important, all right? It provides some protective immunity, all right? So that's, put a star by that sucker. IgG and IgM both have the potential to fix or to activate complement. And they do it slightly differently, but it's similar idea to stimulate the complement cascade. The oftentimes it's the FC region, especially IgG, that's stimulating the activation of complement. It's the complexes that are forming with IgM that stimulate complement cascade because the FC region isn't available. All right. So the FC region, that tail of the Y, all right, it has the potential with IgG to link up to phagocytes. So it serves, the antibodies may serve as opsonizing agents, so coating, covering some sort of foreign invader. And then these would be like little red flags sticking out saying, come get me, all right? The phagocytes recognize these red flags, and there are receptors, FC receptors on the phagocytic cells that will bind to them, causing that foreign substance, that bacterium typically to be engulfed, all right? That's the idea, all right? Doesn't happen with IgM because it's pentameric. All of those FC regions are already tied up. It doesn't happen with IgA because it exists as a dimer. So again, FC region tied up. The IgD, it doesn't occur. The FC region doesn't interact because it's already studded inside or linked up inside of the plasma membrane of the B cell, right? Holding it tight. So it's not able to participate in any sort of reaction. And the last one, this is, well, so either stimulating phagocytic cells with IgG or IgE at the end of the table. This is where we start seeing issues, all right, allergic reactions. The FC region on an IgE molecule stimulates and interacts with mast cells and basophils, receptors on the mast cell. I don't know if the basophil has an FC receptor or not. Matters not, right? They interact with the FC region and stimulate both of these types of cells to release histamine, which in turn stimulates, well, allergic reaction and a response that typically we would say is not so favorable. All right. So there's tidbits about each of these as far as what they do. I've already kind of hit that. And you can see neutralization, that'd be like inactivating a toxin that maybe a bacterium is secreting. Agglutination is the clumping, the pulling together of cells. We already saw some images of that. Stimulating the complement cascade. Opsonization I described as the coating of a substance to kind of make it be flagged or tagged for destruction. And then there's also the stimulation of antibody-dependent cell-mediated cytotoxicity. We showed some images of that a couple of lectures back. Okay, so hopefully that will give you a little bit of an idea as far as what you want to be thinking about. What's the role of a B cell? I just told you, it's right here. So B cells are what's going to be pumping out these suckers, all right? These immunoglobulins. The B cell, upon stimulation, and we will, here's the FC receptors on a macrophage. Here's our bacterium that's already been tagged, opsonized with this immunoglobulin. So we've linked up to it. The FC regions on this immunoglobulin are able to link up to the FC receptor on this phagocytic cell on the macrophage, causes it to invaginate and pull it in, and then we will destroy it within a vesicle. So the B cells, there's more details to the B cell, but truly the idea with the B cell is antibody production. And so we're going to see recognition of antigens, and you can see here B cells with those type 2 receptors as well, where they're going to get in and interact. So And as we progress through, we'll hit some more with the B cell. I do want to show this figure because this is another good one. This is a dendritic cell, but either way, any sort of phagocytic cell could be doing this process. So here, the bacterium gets engulfed. All of the little red bits on this bacterium would be thought of as like the PAMPs, those antigen or, protein, typically protein or sugar glycoprotein rich regions on the surface. It's the outer bits usually that we recognize as being foreign and mount a response against, all right? Rarely is it internal components that we think of as being antigenic, because how likely is it that you're going to be seeing the insides of the bad guy when you're just doing surveillance? Not very likely. You see the outside, right? Those surface proteins or sugars that you may recognize as being foreign, all right? And that's why we're showing it on here all studded up. It's these red guys that become important, all right? So we phagocytize this guy, we recognize it, pull it in, maybe recognizing it's these surface things that are foreign in the first place. We invaginate around it, pulling it in inside of a vesicle. This vesicle fuses with the lysosome, becoming a phasiolysosome. Sometimes they call it a secondary food vacuole. Phasiolysosome definitely sounds more scientific. So here's the little lysosomes. They're coming in, fusing with this, dumping their digestive enzymes on this foreign invader, destroying it. And the red guys, they're the ones we want, right? So they get released, these antigens, and they link up with proteins that are being produced simultaneously, all right? These proteins that are being produced are MHC2 molecules, all right? Sequences of amino acids that are going to basically do the show and tell, all right? So we're making these MHC2s. They're being produced. The antigens are being released and packaged on to the MHC2s simultaneously, all right? There, here we're looking at one thing, right? One little red circle, but there could be a handful of different colors or shapes that could be antigenic, and each of these things could be released and could be packaged up on an MHC2 and presented to the outside, all right? This dendritic cell has the capability of showing multiple cells all sorts of different things, not just one thing, all right? As it becomes this antigen presenting cell, it wants to show everything that could be possibly targeted, all right? That's really what, in a good functioning immune response, that's what you'd like to have happen, right? So the cell would be all studded with MHC2 molecules. We're just showing one of them to simplify it, but they'd be all over, which in turn would flip around and communicate with the T cell. That T cell, in turn, differentiates and decides what sorts of a response do we want to have. A T helper 1 stimulating cell-mediated immunity, a T helper 2 response stimulating humoral immunity. And if we do that, that T helper 2 cell flips it around to a B cell and tells this B cell, this is what I've got. So it shows this antigen to the B cell. And as it shows that B cell what's going on, the B cell produces a similar picture on its B cell receptor, IgD, right? That B cell receptor now looks like the opposite side of this antigen. It's ready to grab it, all right? And then that B cell starts circulating around doing surveillance. And when it finds one of these, it will link up and it will convert into a plasma cell, starting to pump out antibodies. That's really what B cells are trying to do as far as the protective surveillance that they are doing. So here's T cells. I know we're getting to a B cell. Here's some, see, so the T helpers, receptors, and this is that initial. So dendritic cell that we were just looking at, as it processes and presents, here's its class 2 protein with that little antigen, and it shows it to this T cell. That T cell again, dependent upon what's being shown. And this dendritic cell could show all sorts of things, right? So you could have numerous T cells that all are going to, one way or another. Again, cell-mediated, humoral, and we also generate some that lay in wait as helper cells. So when we see this later on, we're already kind of primed to react. That's kind of the idea with that. Okay. So this table differentiates further. It's the one that was a little more detailed than the one I skipped over. The T helper 1s, T helper 2s, these things you don't need to necessarily get bogged down with, but this is important. And there's definitely a question about this, 1s versus 2s. Okay, let's see if I've got anything else. Any other questions as I keep scrolling down here? Anybody that's here live? So silent. It's St. Patrick's Day. Good times. Some have already started celebrating. You shouldn't. Not at 9:30 in the morning. This more with the it looks like an immunoglobulin. This is IgD. This is B cell receptor. All right. So plasma membrane of the B cell. This is the FC region, like I said, linked up into it. So it's not available to participate in any reactions. And the binding site, again, would be respective to whatever that T cell was trying to show it in the first place. It's like, oh, I can do that. And once it recognizes that, those receptors link up to that bacterium in this case. And as soon as it happens, they say, oh, we know what we need to do. And it starts secreting IgM molecules. They, again, are the first immunoglobulins that kind of show up on the scene. Short-lived, we'll have other cells that start secreting IgG. Shortly after, these cells start pumping out IgM, all right? So these IgM molecules, or respectively the IgG that's going to be pumped out a little bit later, the binding sites, the antigen binding sites on these immunoglobulins are the same as the antigen binding sites on the surface of the B cell, right? It's the same signature, it's the same key. It's like a little key factory, pumping these things out so that we recognize the same thing, all right, as we start trying to respond. With that said, that cell, that dendritic cell with numerous types of antigens that are being presented would stimulate numerous types of T cells to produce handfuls of B cells with their respective profile that's going to recognize that type of antigen. And it's likely that you're also going to be presenting stuff that's going to make those T cells say, hey, we should call in some macrophages. They could help deal with this stuff too, and go down the cell-mediated route as well, all right? So just depends. Okay. And that's really what we've got happening here with the B cell and recognition of that proliferation. You generate some memory cells. And this, again, like vaccination becomes important when we think about enhancing the level of protection as you've got more memory cells laying in wait. Lots of them are the plasma cells that are the antibody secreting factories. All right, that's the idea with that. Okay. This is the detail of that figure that we started with. I don't remember if that's where we started or not. But IgM, IgG, and again, where it was averaged together, you've got your first primary response and then the secondary response that's considerably greater. All right. Here it's got some more details. You've got a little further days with this. Again, the first bit, about 2 weeks into it, IgG peaking maybe 3 weeks after that primary exposure. It persists a little bit longer for sure. Then you see the IgM. And then it starts to decline. Here we've got the this saying, there's this gap in time, all right, between the first and second exposure. Typically, all right, maybe not if we're talking a vaccine schedule, it might be, 30 days right after this first exposure, we get another shot, right? Or whatever it is, right? The time frame. And there's definitely differences with that as far as how long you need to wait. If you've got little kids, vaccine schedules seem pretty often, pretty frequent. Once they're a little bit, a month or so, they start thinking, all right, it's time to think about some vaccines. And lots will be administered over what seems like not a very long time, all right? And multiple of those shots require boosters, maybe repeated boosters, before they hit 5 years old and start thinking school age. All right. See if I've got any chats up here. Yes, sir. Okay, that's a good question. So the autoimmune stuff, that is, let me switch chapters. There is a table that is very good with that. Diseases, disorders, I think it's right within here. So here, there's a little bit of detail with each of these. There's actually quite a bit of detail. You don't need all of that. Let's see, where is this table? Okay, so here, what table 19.6 is what you should focus on. And really, this table's got a handful of extra disorders. The ones I posted out on the study guide are the ones I'd focus on, all right? Not that the others aren't worthy, all right? But those are really the ones I'd focus more on. And it's more, do you know, okay, here's the disease. What's it do? It destroys the adrenal gland and some of the issues. Just briefly, weakness or fatigue, something like that. That, move on, all right? So that's the idea. And I think on the study guide, I spelled off what was it, 5, maybe, different diseases. So again, there's a handful of extras on here, but that's it. Okay. And I can talk through some of that if you've got questions about it specifically. Anything else? Okay. No, it's not the same as white blood cell. No. So an immunoglobulin is an antibody, and there are five classes of immunoglobulins or antibodies, all right? We think about IgG and IgM as the two main classes of antibodies or immunoglobulins, all right? And when it comes to those guys, the potential for them to recognize basically anything and everything is there, all right? As long as they get stimulated, there is the potential for that, all right? We're not going through all sorts of details with this. The text does have some extra information, but in the process of generating these cells that are going to be the B cells that are going out seeking the, bad guys, if you will, there's some clonal selection. This happens with T cells, it happens with B cells, where we can go through and recognize these cells are programmed to target bad guys, or these cells are programmed to target good guys. And you don't want them targeting the good guys. So the clonal selection helps to prevent immune cells from recognizing ourself as being foreign, and it helps to prevent autoimmune diseases because you don't want a bunch of antibodies targeting your own self, right? Obviously, there are issues with that because we got this stinking table right here where we've got autoimmune diseases where antibodies start targeting our stuff. That's when this surveillance, this selection process doesn't maybe necessarily function like it should. And then you wind up with some bad guys, right? So as far as antibodies and immunoglobulins, they're the same thing, all right? There is massive amounts of variation with them as far as the variable site, what they can really see. And there's 5 different classes that have the potential to participate slightly differently in an immune response. But the overall idea really is protection, right? Even the IgE that we think about with allergic reactions and bad news, there are some mechanisms. If you're infected with parasites, it does become important. The ill effects that you see are still present as you see histamine and other things being released, but it is a defense mechanism to try to, fight the worms that you may be dealing with or something like that. Does that help clarify it a little bit? Yeah, so the white blood cells, or these types of leukocytes, which are white blood cells, there's all sorts of variation with those guys. You've got granulocytes like neutrophils and basophils and eosinophils. You've got agranulocytes like your T or your B cells. So there's handfuls of different types of white blood cells, and all of those have various functions. The B cell that makes antibodies is a white blood cell, all right? It is one of the white blood cells, but antibodies themselves are not white blood cells. Okay? Yep. So much stuff. Holy cow. Stinking immunology. Okay. Let's see. I'm making sure I'm not skipping one. Importance or role of inflammation, cells and molecules involved. So inflammation, there's good and bad to inflammation. The good stuff we think about stimulating cells to come in and recognize that there's an issue. Maybe thinking a puncture wound or something like that, stepping on a nail and getting bacteria into to your foot. You want cells to recognize there was an issue and to respond, hopefully very quickly. So an inflammatory response helps to stimulate that. You can see vasodilation, where you get increased blood flow, increased circulation, helps to enhance the delivery of some of these immune cells to recognize and help fight off those invaders. So there's, numerous cells that potentially participate in an inflammatory response. Initially, we start thinking about some of these first cells showing up, stimulating it. Platelets releasing factors that stimulate cells to come in, macrophages and neutrophils releasing cytokines to stimulate other cells to come in. There's numerous players, right? We'll also think about the mast cells and the basophils with histamine and other substances that enhance the inflammatory process. Again, stimulating more cells, all right? That's kind of the focus of that. Other stuff. This is good. We're hitting a lot of stuff here. I hope you guys are well prepared. Yes, sir. That's a good question. So what's the difference between cidal and static, like bactericidal or bacteriostatic? I'm scrolling through the study guide. That helps nobody at home. It's one of the first questions that's on there. And this probably was chapter 13 stuff, maybe. So thinking specific substances that you might be using, bactericidal, and it doesn't have to be bactericidal, it could be any sort of cidal. That means to kill, right? So that's going to inactivate all sorts of different processes within the cell. There's different modes of action for substances that could be doing this. You don't need to get bogged down with that. But it causes some inactivation inside of that cell and it kills it, basically. Cytal is to kill. So you think of sterilizing an autoclave is a good example of that, to kill substances. There's other things like your disinfectant that you spread onto the bench tops in the lab. That's a bactericidal agent. If you follow the directions and spread it on and allow it to air dry over the course of a couple of minutes. If you spread it on and just wipe it dry immediately, it's probably not going to be as effective. And in those circumstances, then you might think a little more static. Bacteriostatic or any other organism static. A static situation would be something that inhibits the growth, but it doesn't kill them. It basically just makes it difficult for them to propagate for a period of time, and then the conditions kind of go back to normal and they keep growing. So it slows their growth. And you might think of one example of something like that as something like the refrigerator. That's a static situation. You may have food that's got organisms in it. You pop it in the fridge, it slows their growth, maybe considerably. Kind of depends on the organism, because there's some that putting them in the fridge doesn't matter. But that's kind of what I think with that. Other stuff, we're about out of here. Okay, guys. I think I will quit. I'm not getting anything else on the chat. It is 9:50. That's quitting time. All right. I hope you all have a good day. I will post this here in minutes, so it will be on there and you can review it. We will lecture Friday. We will start new material. So start thinking about pathogens. Not that I ever really have stopped. Okay, good. Farewell. Have a good day, guys. Happy St. Patrick's Day to you.